Good afternoon, everyone. We'll wait for 1 more minute and then we will start. Am I audible?
Yes, ma'am, you're audible.
Okay. I hope my screen is visible. So let's start. So first, topic wise, we will come. So first is what is self-assembly, right? So I think everybody will know what is self-assembly. So it is a self-organization of molecules, and of course there is spontaneity. So spontaneous self-organization of molecules. Okay, and it applies some kind of non-covalent forces which will come later. So these are the some basic examples of self-assembly molecules. So in dendrimers you can see this kind of where molecules, this kind of structures where you can see hydrophobic tail and hydrophilic head are there. So this is the system of inorganic nanoparticles. Then there are array of polymers. These are the proteins. These are the metal organic macrocycles and the other things. Okay. So you will see the self-assembly particles. Here are some other examples. So this is the peptides bonds, and they are amphiphilic peptides, and they self-assemble around some kind of lipid particles or some kind of surfactant, some kind of pore are there. Okay. In living cells also you can see the self-assembly. Okay, so here one example is given that there is some monomers and these blue chains are some stabilizers, and when the polymerization happens, these monomers will form the polymeric chain, and the stabilizing agents will self-assemble around the polymeric functional material, and they will help in form a stable solution, colloidal solution. Okay, so this is the self-assembly. I have given some videos, attached videos, you can see there also, but I am showing one.
Explain what is self-assembly.
So you can see that the proper manner to attain a state or least energy, these components reorient and rearrange their positions. So they are self-assembled. We don't need to put any external forces to self-assemble them. Okay, this is the minimum energy required to self-assemble. Okay. Organized structure or system is formed from a disordered system of components as a result of specific local interactions among the components without any external work. Okay. So you can see that how the raw molecules behave, and now the intermolecular interaction is there, and they form a kind of self-assembled structure. Okay. Okay, so you will see the video later. I will share the slides and you will get to see the video. Okay, proceeding to the next question, that which arrangement is most stable and why? Which arrangement is most stable and why? So in self-assembly, the process will always follow a least interaction energy path where the work done is least. Okay, so that is the that is the most stable arrangement in self-assembly. So here it is written that the one that has the lowest interaction energy. So I have given here an example. So I think many of you know about liquid crystal. So liquid crystal is a material which is the state of matter between solid and liquid. Okay, so you can see here that in solid, the molecules are arranged properly, regularly, in the way it is organized in a crystal, right? And but in case of liquid, there is no molecular orientation among the molecules. They are randomly oriented. But liquid crystal is in between them. So they have some kind of orientational order, but they don't have positional order. Okay, so this is your liquid crystal. Then what happens? So when liquid crystal is in emulsion form, they generally form two kinds of structures. First one is called the radial arrangement, and second one is called the bipolar arrangement. Okay, so when you see this material, when you see this kind of liquid crystal material under microscope, you will see different types of beautiful patterns. Some kind of beautiful patterns. So why this molecule orientation changes from radial to bipolar? Because this actually defines our self-assembly, because we are not forcing any external forces to reorient the molecule. They are self-assembled. Okay, so you can see the self-assembled step of liquid crystal molecules. So initially you will see the molecules will look like this. This is the schematic structure. This is the bright field microscopic image, and this is a image under the polarized microscope. Polarized microscope means there is only one wavelength is passing. Okay, so initially the structures of the liquid crystal molecule is like this, and now the molecules are self-assembled among themselves inside the droplet. So now the structure is little bit changed. Okay. Again, in the third case, you will see it is showing a different, forming a different self-assembly. That is why you are showing, you are seeing this kind of structures. Fourth one is this. Fifth one, the defect you can see here, and at the end, you will see the defect at the middle. So from bipolar to radial, it doesn't change directly. It passes through several steps to self-assembly. So this is one example. There are many examples. Okay. Next is what are the two types of self-assembly? What are the two types of self-assembly? Tell me. You can write in the chat also. What are the two types of self-assembly? So,
Active and passive.
yes, you can say that way also, but as per the literature, if we say it will be static and dynamic self-assembly. Okay. This is the static and dynamic self-assembly. So static self-assembly means, once the structure is achieved, once the final structure is achieved, it will not it will not change anymore. It will be stable. So it will be stable. And dynamic self-assembly means it will change over time. Okay, so it is not stable, but some kind of self-assembly of the molecules is achieved. Next, what drives the self-assembly? What drives the self-assembly? That means what kind of forces lead to this self-assembly of the molecules? What kind of forces? So there are various forces. Mostly weak forces are there. So first one is the chemical interaction. First one is the chemical interaction. Next is physical forces, like you can say physical forces like magnetic force. Then comes the electrostatic force, right? Then there comes the polar nonpolar forces. Polar nonpolar forces. That means that is the hydrophobicity, hydrophilicity kind of thing. Okay. Hydrophobicity, hydrophilicity, like that. Next one is the shape. Shape also leads to the self-assembly around a molecule. Okay, so shape is an important parameter. Next is the templates. Templates means we are forcing the self-assembly. That is, it is self-assembled around a certain pattern. So that is called template. Okay. Next, name some of the chemical interactions. So one of the most important driving force for self-assembly is the chemical interaction. So what are those chemical interactions? We have to specify. So what are those chemical interactions? Tell me one or two names.
Hydrogen bonding.
Yes. Hydrogen bond.
Pi-pi interaction.
Yes. pi-pi interaction. Then comes the Coulombic interaction.
Van der Waals.
Yes. All these are very short-range weak forces. So they lead to chemical interactions. Okay. So now comes the DNA nanotechnology. So first tell me, what is DNA? What is DNA? So you all know what is that? What is DNA? So DNA is the basic molecule or primary molecule in our body. So DNA is dideoxyribonucleic acid, right? So another one, there is ribonucleic acid, but we are talking about DNA here. So DNA consists of some of the adenine, thymine, cytosine, guanine. These are the base pairs, basic base pairs, and it has phosphate backbone. Okay, so you can see this video. The precise structure. So later comes the DNA nanotechnology, but the basic is DNA. This is so you can see later also. Yes, so basically it is the basic primary molecule in our body, which carries information from one place to another place, and it has this basic structure, that is double helix structures, and it has phosphate backbones which have base pairs. Okay, and DNA nanotechnology means we are using this DNA to form different kind of nanotechnology which are applicable in our biomedical research or biomedical machine, medicine, drug delivery. Everywhere DNA nanotechnology is used. Okay. Next comes the DNA origami. So it is the same thing. We are using DNA to make small nanostructures, and we are call it DNA origami. Okay. So in DNA nanotechnology, what are the basic things? Basic things is that it is completely based on the self-assembling branched of DNA complexes with useful properties. Okay, and it has DNA is very much exploitative to use for their unique structure, unique applications. Okay, so these base pairs can be manipulated to use in our different biomedical research. Okay, so next comes what is nucleotide? Nucleotide is also part of the DNA. As DNA is based on DNA is based on nucleic acids, and these nucleic acids are based on nucleotides, right? So what is nucleotide? So nucleotide basically consists of sugar plus base plus phosphate, right? So nucleotide is consisting of sugar, base, and phosphate. And all these DNA and RNA has the sugars, carbon sugars. And sugar and base together is called nucleoside. So nucleotide is a part of nucleic acid. And nucleic acid is a part of DNA. So nucleotide has become an important part of DNA. Okay. Next, how DNA is synthesized. How DNA is synthesized. So we'll see this video. So what is written? Virtually every cell in your body contains DNA, or which carries our gene, which makes different our cells from one another, right? The discovery of DNA is a chemical structure by Watson and Crick opened the door for synthesizing and manipulating genes using phosphoramidite chemistry. Single bases are added, growing oligonucleotides, control pore size, and this large-scale printing of oligonucleotides lead to the formation of DNA. Okay, so this is the synthesis method of DNA in laboratory scale or in industrial scale also. So basically there are few steps, which is that oligonucleotides are there. Oligonucleotides are synthesized using DNA synthesizer. That is DNA synthesizer. Then they are purified. Next step is purification via electrophoresis or chromatography. Next step is mixed together at proper molarity ratio. Next is heat or thermal activity to denature, and then gradually cooled to allow the single-stranded DNA molecules to find their correct partners, right? So if you see from the lecture itself, so here. So it is this. So initially the DNA, single-stranded DNAs are there. When the heat is given, they will find their proper bonds and they will club together. Again, if we reduce the temperature, they will become single-stranded DNA. Okay, and always this adenine and thymine pairs are together, and guanine and cysteine pairs are together. Okay. Next, why DNA has double helix structure? So you can see this structure is called the double helix structures, where there are base pairs ATGC, and these are the sugar phosphate backbone at the both sides.
which are essential.
So this is the deoxyribonucleic acids, and types of nucleic acids are the proteins, lipids, and the phospholipids. So this is your DNA. It has two kind of phosphate structure, or this is the two kind of helix structure. Okay, so it is form of nucleotide. Nucleotide we just have learned, it is the sugar and the base portion. Right, and this is the nitrogenous base, this is the phosphate group, this is the 5-carbon sugar, total called nucleotide. Okay, so now these backbones will interact. So this is the phosphodiester bond. These are the bases in between. This is 4 bases. So they will lead to the sequence. So everywhere you can see always A will pair up with T and G will pair up with C. Sequence encodes genetic information. This genetic information is very valuable, and it is there in each of our cells in our body. So that is differentiating among human beings. Okay. So what happens? Little bit. So DNA transcription happens, it forms RNA, and RNA translation happens, it forms the protein. Okay, so this is pyrimidines and this is these are purines. So question may come that which bases are called purines and which bases are called pyrimidines. They may give some kind of MCQ options. You have to choose. So remember very carefully. See how these bonds are formed inside the DNA. Okay, so you will see that video later. Then comes the ligase enzyme is used to cut the DNA. Ligase enzyme is used to cut the DNA. Is it true or false? Yes, it is false. It is false. Yes, ligase is used to join. Ligase basically joins the DNA, right? So what happens when there are two strands of DNA, different two strands of DNA, they, with the restriction enzyme or hydrogen bonding, they will, suppose these are, this is the single stranded DNA, these are different kind of single stranded DNA. So they will form a structure together upon hydrogen bonding, and now the ligation happens and they will join these two and make a double helix structure. Okay. So this is the ligase enzyme job is to join the DNA, two different kinds of DNA. Next question. How many bases must be paired for a double helix to be stable at room temperature? How many bases must be paired for a double helix to be stable at room temperature? So the answer is about 8 bases. Okay. The answer is about 8 bases are required to form a stable DNA structure or stable DNA double helix at room temperature. Okay. Next, what are the steps of DNA origami? So here also you can see, but I will come to later. Let's first see the video. So this is similar to the paper origami kind of thing that we cut the paper in different shapes and we join them to structure a new thing, right? So DNA is also same like that. So we cut it in suitable positions, suitable points to make them used in different cases as per our biological requirement. Okay, so this is the double helix structures of the DNA. Two groups of strands twisted together, and these strands can be separated. A single strand of DNA without, this is called single stranded DNA. So if it is possible for DNA to unpeel into single strands, keeping those strands together, double helix structure forms. These are individual units are called bases, right? ATGC. Then their pairing is key for the double helix structure. The pairing between the bases is the main thing, the most important thing for having the double helix structures of DNA. See that these are the single stranded DNA are full with the base. Now they will join. Now they will combine with each other and form the double helix structure. See what is written in lab. You can do much more from just forming the double helix DNA. So we have a long single strand of DNA. Now you create short fragments of them that are complementary to the small portions of your strand. Okay. These fragments are called stable bonds to its complementary fragments. You can see how they are making the fragments. So what you did, you take out two portions of your single-stranded DNA and separate it from the double helix structure and form a new kind of pattern or new kind of structure. This is the DNA origami. You can actually manipulate the single strand of DNA to fold into pretty much any shape you want, right? And this technology is used only to deliver drugs in human body, right? So this is the advantage of the DNA origami. Next video you can see later. Next comes our protein. What is protein? What is protein? So it is a large molecule doing mostly all the works inside our cells, right? And they consist of smaller units of amino acids okay, more than 200 amino acids should be there to form a protein. Okay. It has different structures, alpha helix structures, beta sheet structures. The basic thing is they are very large molecules, consist of many numbers of protein, many numbers of amino acids. I am showing these videos from YouTube because then only you can see these things in front of your eyes. The main thing is to do these experiments in the lab, but doing lab is not possible here. So at least we can show the animation through the videos. That is why I am showing. So you can little bit understand what is actually happening and what is the structure of these things. What we are talking protein, DNA, RNA, peptides, then comes the liposomes, surfactants. What are these? How they actually look? You can understand little bit from these videos. That is why the videos are given. Okay, so protein is one of the important things, and this consists of the two amino acids. So these are the amino group. Carbon group, alpha carbon are there, side chain are there. You can see the standard amino acid. These are standard amino acids. Hydrophobic amino acids have carbon-rich side chains which don't interact well with water. Hydrophilic or polar amino acids interact well with water. Okay, so amino acid also can be of hydrophilic type and hydrophobic type. And accordingly, they will interact with polar solvents like water because water is the main part of our body. Charged amino acids interact with oppositely charged amino acids or other molecules. So now comes the protein structure. The primary structure, secondary structure, tertiary structure and quaternary structure. The primary structure of protein is the linear sequence of amino acids as encoded by DNA. So you can see in the video. The amino acids in a protein are joined by peptide bonds which link the amino groups. You can see peptide joins the two amino groups. Okay. A water molecule is released each time a bond is formed. Because what happens? I am showing you. Peptide bonds always join. I think I will come to peptides, right? Okay. So protein molecules are joined. The amino acids of protein molecules are joined through the peptide chain. Peptide chain. It is a small chain, and it joins between the COOH bond of one amino acid to NH2 amino group of another amino acid, and they will bond together. What will happen? They will form a CONH group. So what is happening? You can see that from COH, CO will be there, and from NH to NH will be there. So what is left? H2O is left. So that is why it is shown here that one amino acid to the carboxyl. It is written here that linear sequence of amino acids as encoded by DNA. This is the primary structure. The amino acids in a protein are joined by the peptide bonds, and these peptide bonds which link the amino group of one amino acid to the carboxyl group of another. So that is what I have shown here. So this COOH comes from one amino acid, and this NH2 comes from another amino acid, and when they are bonding together, they release a water molecule and form a CONH bond, and this bond is called the peptide bond. Okay. Carboxyl. Yes. So a water molecule is released each time a bond is formed, right? The linked series of carbon, nitrogen and oxygen atoms make up the protein backbone. So you know that already. And now comes this is the linear chain. Basically, primary structure of protein means this is the linear chain. Now secondary structures is, they often fold into two types of secondary structure, alpha helix and beta sheet. So this is the alpha helix structure. These are the beta sheet structure. Okay. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl group. Okay. And beta sheet are formed when the hydrogen bonds stabilize two or more adjacent strands. What happens? Okay. Again, it is coming. I think you are able to see here. The call will end in 50 minutes. So again, I am sharing a new call. So I am sharing the new joining link. As you can see that it will be ended in 15 minutes. So please join this new link and then we will start in the new session. So everybody please join the new link I have shared in the group. Share in the chat, sorry. So please follow this new link. So this call is going to end in 5 minutes maybe. So please join the new link I have shared in the chat. I'm ending here. Okay. Yes, I think now I am audible because I was talking to the other session. So now I think this okay. Yes. So where were we? We were learning the protein. So next comes the tertiary structure. So tertiary structure of a protein is the three-dimensional shape. From the name itself, you can understand. Linear means the basic linear structure, primary. The secondary was 2D shape structures. Tertiary means the 3D structure of the protein chain. Right? So everywhere the material is same. There are everywhere there are charged amino acid, hydrophobic amino acid, hydrophilic amino acid, like that. Okay. Many proteins form globular shapes with hydrophobic side chains sheltered inside, away from the water. They can interact with lipids. Charged amino acids allow proteins to interact with molecules that have complementary charges. which is the normal case, right? For example, hemoglobin forms a pocket to hold the heme, a small molecule with an iron atom. What happens in the center that binds the oxygen? This is the example of tertiary structure of the protein. Next comes the quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. So the four subunits of hemoglobin cooperate so that the complex can pick up more oxygen. Okay, so more than one peptide chain are there. Okay, so these are the four structures of protein. So you can see here this space is filling. So here you can see there are more than one peptide chain, peptide bonds are there to carry the heme. Right? So these are the different types of diagram. The protein size is 6.5 nanometer. Yes, a typical red blood cell 6.5 nanometer means how you cannot even visualize that length scale. So a typical red blood cell contains about 280 million hemoglobin molecules. Just imagine the vast mess. So now 280 million of hemoglobin means almost 10 micrometer size, right? So, these are the advantages of using protein. That protein does what basically? The arms of antibodies protect us from disease by recognizing and binding to pathogens and target them for destruction by the immune system. This is the defense mechanism of protein. This is the communication mechanism. So this is the insulin receptor. Hormone insulin is small, stable protein that can easily maintain its shape while traveling through the blood to regulate the blood glucose level. So it is used as a glucose transporter. Protein enzymes are also one kind of protein, okay, like alpha amylase. So alpha amylase is an enzyme that begins digestion or starches in our saliva, right? So it breaks the starch bond, okay, and that generates the glucose level, that controls the glucose level in our body. So I was telling this, so everything inside our body is protein. So this is protein works as a defense. Protein works as communication. Protein works as enzymes. Protein works as transporter. It is used as storage and structure. So that is what we are. I am sharing. Maybe the call was cut because of the network. So this is the defense mechanism. It attacks the pathogens. In communication of the insulin, it works. regulate the blood glucose cells. Then comes the enzyme. Enzyme is the alpha amylase. So alpha amylase breaks the starch level and helps in digestion and produces glucose. Enzyme acts as a transporter in muscle cells. So it transports the calcium and magnesium to generate ATP. Next, how it stores. So ferritin is a spherical protein with channels that allow the iron atoms to enter and exist. On the inside, ferritin stores iron in the non-toxic form. Okay, so this is the advantage of protein. Collagen structure is also protein. Okay. So collagen molecules can form elongated fibrils which aggregate to form collagen fibers of 0.1 micrometers. This type of collagen is found in skin. Okay, so now you can understand how multi-purpose proteins are in our body. Okay. Next, the first member of peptide Lego was discovered from where? The first member of peptide Lego was discovered from where? You can answer in the chat box also. The first member of peptide Lego was discovered from a segment in the left-handed ZDNA binding protein in yeast, and it was named as zootin. That means the peptide bond is first discovered also in the DNA, because peptide bonds forms the proteins, and protein is formed from the protein is synthesized from the DNA. Next question, what is Pura matrix? What is Pura matrix? Anybody knows? Which one is Pura matrix? So Pura matrix is this. This is a peptide scaffold. This is one kind of peptide scaffold. Okay. Next question is, what is K2V6? So in peptide bond wheel only you will find the structure K2V6. What does that mean? K2V6 means, so you can see K2V, how we'll write K2 and 6V. Okay. So to positively charge lysines as the hydrophilic head, followed by 6 valines as the hydrophobic tail. This configuration is called, this is also a configuration of the self-assembled configuration of the peptide bonds, and This is the K2V6 structure. Okay. Now let us do some question answer. Maybe for the old assignment, I will do first, then I will come to the new assignment. Okay. So you have to just tell me. Consider the following: ionic interactions, non-covalent interactions, van der Waals forces, hydrogen bonding, magnetic forces. Which of the above mentioned forces play an important role in the process of self-assembly? So we have just gone through the self-assembly portion. So I think you will be able to tell that which forces act when we talk about self-assembly. Please respond in the chat. Only option 3, 2, 3 and 4. 2 is non-covalent interactions, van der Waals interactions, hydrogen bonding. Yes, 2, 3, 4 are there obviously, but it is also the ionic interactions and magnetic forces, because once I have showed you there that what are the interactions here. So in the physical interactions, there are electrostatic interaction and magnetic interaction. So magnetic forces will also come in this section. Okay. So
Ma'am,
yes.
the question was, do they play important role?
Yes, magnetic force also play important role sometimes, but not always. Obviously, the non-covalent interactions and van der Waals forces are mainly functional here, but sometimes magnetic force can also be applied. Well, can play a role. Okay.
Okay, understood. Okay.
So we have to go for all 4 because as the option is given here, so it will be all of the above. Next is the Chargaff's rule in DNA nanotechnology implicates what? This is very easy. The Chargaff's rule in DNA nanotechnology implicates what? So, like, what are the basis, pattern, or pair which will apply on DNA molecule?
A to T and G to C.
Yes, it is always A, T, and G, C. Next, alpha helix and beta sheets are terms associated with what? Alpha helix and beta sheets are terms associated with what?
Structure of DNA.
No, structure of protein.
No, protein.
Yes.
Protein, yes, ma'am. Sorry, protein.
Structure of protein. Next is the charges of cationic peptide tubes and DNA are negatively charged or positively charged. Positively charged and negatively charged, both are positively charged and both are negatively charged. It is saying that cationic peptides are like what type of charge is there in cationic peptides and what type of charge is there in DNA.
Ma'am.
What?
Second option.
Yes, it is the cationic peptide is positively charged and DNA is negatively charged. Next one is the liposomes are spherical vesicles. Okay, liposomes will come later because I have not gone through it. Okay, so liposomes. Maybe we'll now do the new question which is given in this assignment. So this is your assignment, right? So what? Select the correct list of lipids. Select the correct list of lipids. Okay, have we gone through lipids yet? No. Which one can do now? Which of the following is the characteristic? This one. We will come to later in the question number 1, but first do the question 2. Which of the following is not the characteristics features of self-assembly?
Tends to create defects.
Yes, this one is the 4. Tends to create defects. Molecular model of lipid-like peptides A6D can be read as the lipid is left. DNA. The effect of salt concentration in solution on DNA structure are defined as the effect of salt concentration in solution on DNA structures are defined as high salt leads to aggregation of structures, low salt concentration leads to aggregation of structure, decomposition of DNA structure in low salt concentration, and decomposition of DNA structure in high salt concentration. What do you think?
Decomposition in low salt.
Decomposition in low salt, yes. Then?
Yes. And high salt and 1 and 3, ma'am.
Yes, 1 and 3.
Ma'am, one more thing.
Yes.
Yesterday we had the same class, ma'am.
Yes.
So in that, ma'am finished this all of the week 2 assignment.
Okay. So, but maybe there are some new students joining in the today's session, so I have to go through again.
Yeah, ma'am. I have a doubt in BZ transition, ma'am. The 5th question. Can you explain that to me? That's what I wanted to ask you.
Okay.
Actually.
Okay. Tell me. Choose the correct
The
statement.
fifth question.
Yes.
Fifth
Yes.
question. The same.
The BZ transition is based on the change in the nitrogenous base pair sequence in the presence of helicase enzyme. BZ transition occurs by changing the ionic strength of medium. Freight used to study the energy transfer between two light-sensitive molecules. Fluorescence quenching is observed in zDNA form of bzDNA motor. Okay. So, which statement is correct? You said, na? So, what is your answer?
Except 1 and 2, I think 3 and 4 are correct answers, ma'am. But they are like yesterday, ma'am said 1, 3, and 4, the second option. But I feel like 3 and 4 is the correct answer. I didn't understand 1 and 2. That's the problem.
Okay. So, let's go to the topic. This is in lecture 9, I think, right? Yes, 3 and 4 is correct, but let's find between 1 and 2. BZ transition occurs by changing the ionic strength of medium. BZ transition is based on the change in nitrogenous base pair sequence in the presence of helicase enzyme. Why do you think that 1 and 2 are not correct?
Usually, is the helicase, should it be helicase, ma'am? That's what I want. PZ enzyme, should it be helicase? Is it required?
Because
It's not specifically required, no, ma'am. That's what I think. But I don't know. That's why I had a doubt. Yesterday, I didn't get the chance to ask. I
okay,
actually
okay.
asked. That's all.
I think helicase will work here because let me search what helicase does. So helicase is a class of enzymes that are vital to all organisms. The main function to unpack an organism's genetic material. Helicases are motor proteins that move directionally along a nucleic double helix, separating the two hybridized nucleic acid strands via the energy gained from ATP hydrolysis. Okay. So there are many to separate strands of DNA double helix or cell assembled RNA. Okay, so helicase is used for separate the strands of DNA double helix. So for that case, I think here also if from BZ to BDNA to zDNA transition, there is orientation change of the nucleic this base pairs, like. So I think helicase will work here. Could I convince you?
Okay, I still didn't get the idea, ma'am, actually. Is it like, why is it exactly we require helicase, ma'am? Excuse me,
Yes,
ma'am.
yes.
Even when I'm searching, I am not getting the same thing, ma'am, actually.
Okay. So
It's like saying it's not based on change on helicase, like that's what they are saying, ma'am.
so what is like how B zDNA is transformed to zDNA? That's the question. How bDNA is transformed into zDNA. Hmm. So bDNA transforms into zDNA through a series of molecular changes, including the alternating purine-pyrimidine sequences, which promote the transition under certain conditions like high salt concentrations or negative supercoiling. Okay, then on the DNA sequence. So helicase, does helicase not used for changing the DNA sequence?
It's required, ma'am.
So from that perspective, I think because in B to Z DNA also the orientation or the ATGC bonds, the bases will be like shifted little bit or alternated little bit, right? So I think to change the sequence, helicase is required here.
Okay, ma'am. So if you want to change the presence of helicases you're telling me the presence of helicases are required
Yes, yes.
for
What
the.
I think, because it is changing the sequence of the molecule also from BD. The base molecules are the sequence of them is changing, right? From BDNA to ZDNA. So that is why I think helicase is important.
But how exactly is helicase promoting that BZ transition, ma'am? Here they're asking
Hmm.
the BZ transition change is based on the helicase enzyme.
Hmm.
Like it's like nitrogenous base pair is required, but not the helicase structure. That's not what is important here, no, ma'am. That's what like I had doubt.
Okay. So I might not have that depth knowledge because I have.
Yeah, yes, ma'am.
Hmm.
The thing is, I searched. I also don't know,
Yes,
ma'am. I searched in the net
yes,
also.
yes.
They are saying the statement is wrong. But yesterday, ma'am said this statement was correct. So I just had a doubt. Otherwise, no issue,
Okay,
no
okay.
issues.
But
Like when it's like, could you please copy paste the first statement and search whether in the net, ma'am? You will see that exact thing.
this one.
Yes, ma'am. Yes, ma'am. And copy paste and just search in the Google. Is it true or not? That's it. I just did it, ma'am. I got doubt. That's why.
Reversible change between a right-handed and left-handed form of the DNA helix that is primarily driven by sequence and supercoiling, not the actions of. Okay, okay.
Action of
Yes,
helicase
yes.
enzyme. That is the reason I had
Okay,
the
okay.
doubt now
So
actually.
I will start little bit and maybe tell you in the next class because I don't know about the helicase much, so I am not able to answer the question rightly. And in helicase, but from the what is written that helicase is required for breaking the hydrogen bonds and or shifting the uh, base pair. So that is why I thought it is maybe right. But here it is directly said it is wrong. So I have to search again. Okay. So for the time being, you can mark 3, 4 also. 3, 4 only. Okay. If it comes incorrect, so I then we will see what to do. When is the submission of this assignment?
10th, ma'am, I guess. Just a second, let me check, ma'am. It's 10th, I guess. Yes, ma'am. It's on 9th, ma'am. Sorry.
Ninth. Okay.
Sorry, 3rd. Not summer. 3rd. I'm really
Okay.
sorry.
Okay.
3rd
Third.
September.
Next, before next week. So or I am giving you the mail ID. You just text me for this. I will find it or we'll discuss. Okay, over the chat, over the
Yes, ma'am.
mail.
Yes, ma'am.
I am just sharing
Yes, ma'am.
my mail ID from
Okay,
in
ma'am.
the chat.
So for now, should I keep 1, 3, 4, ma'am, options or 3
For
and
now,
4
you
is
keep
enough?
1, 3, 4. Then we'll discuss.
Okay, ma'am. Thank you.
I am giving my mail ID in the chat box. Okay, so you just like send a mail, then I will reply back. So we'll connect them. Okay, so proceeding to the next question. I think 1 and 3 is left, so we'll do that. Select the correct list of lipids. The first question is select the correct list of lipids. So it is obviously the option 3. Fatty acids, glycerol, cholesterols, and waxes and triglycerides. Right? In the question number 3, molecular model of lipid-like peptides A6D can be read as what?
A is alanine and D is aspartic acid. So we can assume that entire alanine to aspartic acid.
Alanine to aspartic. Option B. Aspartic acid is negatively charged. Yes. Oh, alanine to aspartic. Option A. Sorry. Yes. Then comes question number 6. Statement A is proteins, ions, DNAs can be used as a nanomolecular communication carrier in living organisms. And statement B is communication distance in molecular communication is measured in nanomicro scale. Which one is true? So here I think proteins, ions, DNAs can be used as nanomolecular communication. Obviously, these are the basic parameters of our life. So they are used for nanomolecular communication. And statement B, communication distance in molecular communication is measured in nanomicroscale. So both A and B are correct. Non-solubility of carbon nanotubes in aqueous medium can be resolved by functionalization of carbon nanotubes by adsorption, functionalization of carbon nanotubes by electrostatic interaction, functionalization by covalent bonding. So I think it is all of the above. It is all of the above. Next is match the following question. So arc discharge method. Arc discharge method, chemical vapor deposition. So we'll go do it like way. So photolithography is related to patterning and etching, right? So A's will be 3. Okay, A's 3. Then comes the arc discharge method. Arc discharge method uses long bundles of tubes with 520 microns. Okay. So P will be 1. Option 3. P will be 1. Next is the chemical vapor deposition uses metal catalyst and inert gas. Right? So Q is 4 and laser ablation used short tubes with diameter in nanometer range. So it will be option 3. Okay. Lipid-like peptides can work as a similar to chemical surfactants, lipase, DNase, and or ligase. It is chemical surfactant because lipid-like peptides have hydrophobic head and hydrophobic tail and hydrophilic head. Similarly, the chemical surfactants also have hydrophilic head and hydrophobic tail. Last question is DNA denaturation occurs by cooling and breaking of H bond between purines and pyrimidines, heating and breaking of covalent bonds, heating and breaking of H bonds, cooling followed by heating and breaking of covalent bonds. What is the correct answer?
Heating and breaking of hydrogen bonds between purines and pyrimids.
So it is the heating and breaking of H bonds. Okay, so I think it is, the questions are done for for this one. We will do some old assignments. Okay, so what is left here? 1, 2, 3, 4. Let's do 5. Consider the following statements. Liposomes are spherical vesicles made of lipid bilayer. Liposomes can carry only hydrophobic drugs and not hydrophilic drugs. Which of the above statements are incorrect? Which of the above statements are incorrect? So we know about liposomes, right? Liposomes is bilayer, spherical vesicle. Suppose this is the spherical vesicle, and inside there is water portion. So hydrophilic molecules will be like this. They will be more congested. Here I am just drawing like this. And there will be another layer where they will form like this, right? So this is your liposome. So the first question is, first line is correct, that liposomes are spherical vesicles made of lipid bilayer. But the second is, liposomes can carry only hydrophobic drugs and not hydrophilic drugs. This is not true statements, because like in the middle portion of this case, in the middle portion of the water layer, they insert our important drugs. It may be hydrophobic or hydrophilic. Okay, both will be carried by liposomes. So only the one is, only two is, only two is incorrect. Okay, so here one is correct, two is incorrect. Next question. That liposomes that typically range in size between 0.05 to 10 micrometer are called as what? Liposomes which diameter is in between 0.05 to 10 micrometer are called what? So 0.05 to 10 micrometer means it's the bigger, comparatively bigger size, right? So it is the
multilamellar.
Yes.
Multilamellar
Yes,
vesicles.
it is multilamellar vesicle. Next is, choose the correct sequence of flow of information in a mammalian cell. So this is very easy. So DNA first, then transcription happens, which will lead to RNA formation, and then it is the translation happens, which lead to protein formation. So it is option C. Next question. The phenomenon of energy transfer between two light sensitive molecules. The phenomenon of energy transfer between two light sensitive molecules is known as FRET, fluorescence resonance energy transfer. Graphene is mainly made up of octagonal, hexagonal, pentagonal or heptagonal rings of carbon.
Hexagonal.
Yes, it is hexagonal. Pi stacking in the context of functionalization of the carbon nanotubes is associated with pi stacking in the context of functionalization of the carbon nanotubes associated with the non-covalent exohedral functionalization. Okay. I have some other questions also which will be doing later. First, just go through the sections where we have left. Just revise all the things, and then we'll come back to the question again. Next question was about the peptide nanotubes, that how peptide nanotubes are formed. So these peptides, we have just understood that peptides are the bond between the carboxyl group of one amino acid to the amine group of the another amino acid. So these bonds can be cross-linked and they will self-assemble. Okay, and now they will form the bond with each other and form this kind of shape. So this is the self-assembled nanotube structures. Okay. Next is what are the important regions of peptide nanotubes? What are the important regions of peptide nanotubes? It is the ligand, linker, and anchor. Right? So as it is written here, so important bonds are important part is ligand, anchor, and linker. So what does they do? Ligand for specific cell recognition and attachment. Okay. So a ligand will specifically attach to some molecules. All ligands will not attach to all the molecules. Okay, so this is for specification, we use ligand. Then comes linker for physical separation from the surface. Okay, so suppose like in case of gold nanoparticle, like suppose there is, I am giving the example of gold nanoparticle, but you can imagine it through peptide bond also. So there is gold nanoparticle. We give some kind of linker here to attach some foreign molecules around the gold nanoparticle. Okay, so these are called linker. Next is the anchor. Anchor is for covalent attachment to the surface. That means it will form a kind of physical bond with the surface. The ligands might be of the RGD, that is arginine, glycine, aspartic acid motif, that is known to promote cell adhesion, or other sequences for specific molecular recognition or specific cell interaction. The linker is usually a string of hydrophobic amino acids such as alanine or valine. Okay, so these hydrophobic amino acids will separate the surfaces. And the anchor can be a cysteine residue for gold surfaces, aspartic acidic linking on amine surfaces, or lysine linking on carboxylic surfaces. Okay, this is the peptide ink region. These are. Next is, what is glyconanoparticle? So you know that glyconanoparticles have a nanoparticle core, and it is like functionalized with some kind of enzymes like glucose or sucrose kind of things. Okay. So it has a core of nanoparticle, any kind of nanoparticle it may be. Suppose I am writing AUNT, and it has coated with some kind of glucose particles or glucose enzymes. Okay, that is called Glucose means any kind of starch or any kind of carbohydrate, you can say. That layer will be around the nanoparticle core. Okay, so this will be carbohydrate or sugar, also you can say sugar coating will be there. That is called glyconanoparticles. Okay. Next, so here in our lab, seniors have done some, like they have formed gold nanoparticle using some kind of enzyme. So here in this work, I have shown last week also. So using the starch enzyme, they have formed gold nanoparticles. So what will happen that this gold around the gold nanoparticle, a layer of starch will form, and that will lead to our glyconanoparticles. Here, similarly, here in the gold around the gold nanoparticle, amylase functionalization happens. So this is also another kind of glyconanoparticle. This is starch, this is amylase. Next comes our lipid. Like what are lipids? So lipid, just now we have said that lipid is kind of chemical surfactant. It has a hydrophilic head and hydrophobic tail. Okay. So from the examples also you can see. So when there are two hydrophobic chains, that is called phospholipid. When there are three hydrophobic chains, there are triglyceride, and some cholesterol waxes are also called lipids. Okay, so around in our cell also there is phospholipid bilayer. Okay, liposome also has a phospholipid bilayer. Okay, so this is the phospholipid bilayer, and you can see that hydrophobic chains are interacting with each other, and that is why this hydrophilic heads are away from the solution. So you can see this hydrophilic head is consisting of phosphate group. Here are glycerol groups, and these chains have saturated fatty acid and unsaturated fatty acid. Okay. Next is liposome. So this is a small spherical vessel of the same phospholipid bilayer. Okay. The structure is same, but it has two layers around the aqueous medium. Okay. So suppose there is a liposome and molecular association is happening. So hydrophilic molecules are inside it. Charged molecules, hydrophilic molecules will be in the aqueous phase, obviously, which is in the middle, because in this middle portion water is there, or you can say some kind of aqueous phase is there. Next, the charged molecules will be around the hydrophilic head portion, because hydrophilic head means they have some kind of charge, some kind of positive charge, and hydrophobic head means they are just alkane chain, right? Hydrophobic long chain. So obviously the charged molecules will be self-assemble themselves around the hydrophilic head, and the hydrophobic molecules will be on top of the hydrophobic chains, because they are attracted to the hydrophobic chains, because these are the hydrophobic molecules. Liposomes can be be modified, so they can be made charged liposome, pegylated liposome. Pegylated liposome means neutral liposome. Okay, so here you can see these are charged liposome. Okay, so and here Their pKa means neutral, means zero charge. Okay. These are immunoliposomes and these are environment sensitive liposomes. So in which cases liposomes can be used? In which cases liposomes can be used? So conventional liposome can be used for drug delivery, positively charged lipid carrier, negatively charged lipid carrier. Okay. So this is the conventional liposome. Liposome can be used, pegylated liposome means just now I have said that they are made neutral. So they have no charge here. In this pegylation means no charge. Then third one is the theranostic liposome. That means liposomes can be functionalized with our analyte, some kind of using the ligand, and now we can attach our target molecule here, and now it can sense it and detect it. Right. So this is the theranostic liposome application. This is the ligand targeted liposome. Here also same thing is happening, but here they are using different kind of ligands like protein, antibody, peptide, carbohydrate, any kind of small molecules. Okay. So these are the various types of liposome. Classifications of liposome. So just a minute. So these are various types of molecules. I will come to that. First, let me write what are the types of liposomes. So this is the SUV, LUV, GUV and the multilamellar vesicles. So basically they are in, first one is the SUV, that is small unilamellar vesicles. Then comes the LUV, large unilamellar vesicles. Then comes GUV, giant unilamellar vesicles. This is small, large, this is giant. Now when multiple these lamellar layers are together, this is called multilamellar vesicles, right? So this is multiple. So this is what is shown in this picture, that this is a unilamellar vesicle or unilamellar liposome, you can say, where there is a hydrophilic pore or hydrophilic heads, and outside there is hydrophobic chains. So when this layer is single, it is called and have small size. This is called small unilamellar vesicle, and the range of this liposome is 30 to 100 nanometer. Second one is little bit larger, where the diameter of the liposome is greater than 100 nanometer, and when this diameter is even more big, it is called the giant unilamellar vesicle, and the size is more than 1000 nanometer. Okay. Now suppose this layer is engulfed between another layer, and in between two layers there is aqueous phase. So this is called vesicle. This structure is called vesicle. Okay. This is the oligolamellar vesicle. The size is between 100 to 1000. This is multilamellar vesicle. Size is greater than 500, and this is the multivesicular. So here you can see there are two set of phospholipid bilayers. So here there is multiple layer. You can see there are so many layers. So this is called multi-vesicular lipose. Okay. Next is, what are the three methods to purify the drug-entrapped liposomes? What are the three methods to purify drug-entrapped liposomes? So first one is very easy, that is the centrifugation. Second one is the dialysis. And third one is the gel filtration chromatography. So you can see here, first one is the centrifugation. So you can see that it is rotating at a particular RPM. It has centripetal force is applying. The particle is moving following this radial path, right? And this has an axis at the bottom. If you consider a side view of this, suppose this is there is an axis of that, and it is rotating in direction way and at a very fast RPM. So what will happen when it will rotate at very fast RPM and a high centripetal force will be applied on the particles inside the solution? So what will happen? They will be settled down at a corner, and they will, like you can see here, they will form a pellet here, and the rest of the liquid portion will be separated from this solid portion. And this is called the supernatant. So this is supernatant portion. So we will be removing that and we will get our desired pellet or desired liposomes, what we want. Next one is the dialysis. So here you can see that this is the phospholipid solution where drugs are entrapped. First it is evaporated using a solvent. So what happens, most of the solvent portion or water portion will be evaporated and thin lipid film will be stayed at the bottom, right? First, hydration with buffer A. So it will, what will it lead to? It will lead to film dispersion. So film will be dispersed when we'll hydrate with the buffer. And then extrusion will happen, and size-based chromatography, using size-based chromatography will separate the liposome. And in the another case, lipid dissolved in detergent plus buffer A. This is the dialysis membrane. This is the dialysis buffer lacking the detergent. It will extrusion, and then also extrusion can happen, and we'll get a solution along with the liposomes, and it will be separated by the size-based exclusion chromatography. Gel filtration chromatography is also similar kind of thing. There is a column of chromatography column, and here there are gel beads and some solvents is there, and this is our sample along with the solvents. Now that will be separated through the chromatography, and it will be, first the green portion will be separated out, next the this violet portion will be separated out, and next the orange portion will be separated out, and we will get our desired lipose. So these are the basic three methods of separating liposomes. Next is what is neosome. What is neosome? So neosome is also similar kind of liposome, but here they are using non-ionic surfactant bilayers. So in case of liposome, In case of liposome, there is phospholipid or lipid bilayers, but in case of neosome, there is non-ionic surfactant bilayer. Okay. So now comes the self-assembly of the phospholipids. So you can see how micelles are formed, right? They are just forming in the aqueous solution. The surfactants will form a chain kind of thing. So this is the micelle. When there are particular core or particular dispersed phase in the solution, and the molecules will be self arranged or self assembled around that dispersed phase in a solution. This is called the emulsion. Next comes our solid lipid nanoparticles. Next comes nanostructured lipid cells. This is the phospholipid bilayers of liposomes. Okay. This is the PDCs. So this is the different kinds of self-assembled phospholipids they are showing. So this is some of the examples. So when there is hydrophilic, so it is written that formation of micelles on the addition of single chain phospholipid to a solvent. So there is one hydrophilic head and one hydrophobic chain, and they will, when they will self-assemble, they will form micelles. Next one is the formation of vesicles on the addition of phospholipids containing two chains. So when there are two hydrophobic chains along with the hydrophilic head, that will form a vesicle. And this will just show that how the size of the hydrophobic chain and shape of the hydrophobic chain will lead to different kind of self-organized structures. Okay. So these are the alternative forms of DNA. So that A, B, Z DNA. There is a video here. This is the, let me check structure of the done. So this is the B form. Okay. It's a very, let's start. B form is a right-handed helix. It is present in very humid environment, around 92% and low concentration of ions. And this is the ideal environment for BZ DNA formation. Okay. Right-handed helix. Most common form are the B form of the DNA. Yes. Let's count 1, 2, 3, 4. This is the 10 base pairs. They are counting that each turn there are 10 base pairs, and there are low concentration of ions. So this is the B form. We look from at the top, you can see that this blue portion is basically the sugar and phosphate backbone, while the yellow or greenish portion is the base, that is the nitrogenous portion. They are perpendicular to the axis. The hydrogen bonds formed between the bases will angle here, and that is called tilt. Base tilt in B form is 6 degrees. The length of each turn is approximately 34 angstrom, The distance between the two base pairs like this is the first base pair and this is the second base period obviously, and they are called helical rise. That is called helical rise. Helical rise is of 3.4 angstrom. There are two groups here. One is called major group and one is called minor group. So this is the structure of B DNA. A DNA is also right-handed helix DNA. If we put a weight on form B and it is expanding on the side, so the diameter of A will be wider than form B and the length of form A will be shorter than the form B. Okay. Now it is present in dehydrating environment and in the presence of ions like Na+, K+ or Cs+. If a cell is facing a dehydrating environment, in order to survive, the B form can take a form A in that condition. Now the number of base pairs present in each turn in A form is approximately 11 base pairs instead of 10, which was in case of B DNA. These are the basic differences. And the diameter of A form is 26 angstrom. The diameter of A form is wider than B form and length of each turn is approximately 28.6 angstrom. The base tilt is approximately 20 degrees, right? So you can go into the details to see the differences. Next comes the Z form. So what is it? Quite wide enough in A form, last but not least the Z form. Z form is a left-handed helix. So B form and A form both are right-handed helix, right? But Z form is a left-handed helix. And it becomes longer. It is a kind of slim kind of structure. They have a zigzag sugar phosphate backbone running in anti-parallel direction. That is why they are called Z form. Alternating purine pyrimidine sequence such as GC, GC, GC. Now the B form of DNA can take this Z form of DNA if the number of proteins can bind to it and in one helical conformation and force the DNA to adopt a different conformation. You can see that presence of proteins that can bind to DNA in one helical conformation and force the DNA to adopt a different conformation. Here in singleton there are 12 base pairs. The distance is approximately 44. So these are the angular distance or the base pair to pair distance, which is different in each case. Right. So you can see the details later also. Okay. Next comes the protein synthesis process. Protein synthesis process is just that I told earlier. You can go through the video also, that DNA transcribed into RNA and that transcribed into protein. Translated into protein. So you can go through the video later. So these are the basics of carbon nanotubes. Okay, so what is carbon nanotube? Basically, there are graphene sheets, right? And they will roll to form a carbon nanotubes. carbon sheet, they will form when they will roll when graphene sheet will roll, they will form a tube kind of thing, right? So that is called carbon nanotube. Go through the video on your own. Then comes the synthesis process of carbon nanotubes. So there are three basic synthesis process. One is the arc discharge method. Second one is the CVD, chemical vapor deposition method. Chemical vapor deposition method. Third one is the laser ablation method. Okay, so these are the diagrams along with it. So in the arc discharge method, two graphite anode and cathode are given, two graphite rods are given, and at inert gas is given, and at the high temp when the power is supplied, this graphite will form carbon nanotubes. Okay, in the CVD method, methane gas is passed through the chamber, and along with the methane gas, there are inert gas also, and at the very high temperature, that means 500 to 1000 degrees centigrade, this will form CNT. Okay. This is a laser ablation method. That means here, this is the graphite target, and we are bombarding the graphite with a particular laser in the inert gas atmosphere in a furnace, and that will form small nanostructures of the carbon nanotubes. Okay. Yes. So I have some more questions. We can do that. So let me write. So first question is the notable characteristic of self-assembly process. The notable characteristics of self-assembly process. So option 1 is they require external energy input. So they require external energy input. Option 2B is they always result in structures far from thermodynamic equilibrium. So they always result in structures far from thermodynamic equilibrium. Third option is they tend to incorporate defects and irregularities. And the fourth one is they occur spontaneously and often approach thermodynamic equilibrium. Right, so it's better to bring the question. So, suppose this is your question. Okay, the notable characteristic of self-assembly. You all know that self-assembly doesn't mean any external energy. They are always, the structure they achieve, it is always in thermodynamic equilibrium. And so option 4 will be correct. Option 4, right? They occur spontaneously and often approach thermodynamic equilibrium. Next question. The typical composition of the linker region in peptide ink is a, you can tell, the typical composition of the linker region in peptide ink. So in the peptide ink, in the linker region, we have studied that it is a string of hydrophobic amino acids like alanine or valine, right? Next question is, so this is a similar question. Today also we have done. In today's assignment also it was there, that molecular model of lipid-like peptides A6D can be read as peptides have 6 hydrophobic alanine residues from the N terminus followed by negatively charged aspartic acid residue. 3A. Next, technique used for capturing specific target proteins and mapping their binding epitope containing peptide. Techniques used for capturing specific target proteins and mapping their binding epitope containing peptide is the Linz technique. So you can go through the peptide portion. It is there in the notes. Question number 5 is, so select the correct sequence for the DRV technique. Freeze dry until complete removal of water. Prepare empty small unilamellar vesicles. Rehydrate gradually. Initially, add a very small volume to form drug-loaded SUVs. Mix with an equal volume of the solution containing the material to be encapsulated within the SUVs. You may answer this question because I am answering the question only. So what is the first step? So if you're there in the meeting, you can answer the question that what is the first step of the DRP technique? First step is prepare small empty cell. Prepare small empty SUVs, that is small unilamellar vesicles. Second one is mix with an
Equal
mix with
volume.
an equal volume.
Equal volume.
Then freeze dry, and then is the rehydrate.
Freeze dry, last
this is the
rehydrate. 1 4 2.
2 4 1
Yes.
3. Next question is following match. So match the following. Multilamellar vesicles. I think the formation of multilamellar vesicles are given here. So which one is correct? So multilamellar vesicles means obviously the multi phospholipid bilayer, right? Small unilamellar vesicle is this range, extrusion. Large unilamellar vesicle, okay, no. Small unilamellar will form this one, sonication, yes. Large
Unilamellar vesicles
extrusion,
will
yes,
form
and
extrusion.
giant one is the evaporation.
Yes,
Yes,
first
so
one.
this is the first one. No, P3, Q2, R4. This one. Next, the question is that statement A is proteins, ions, DNAs can be used as nanomolecular communication carrier in living organisms. Statement B, communication distance in molecular communication. This one is both are correct. We have done this. Both A and B are correct.
You picked the wrong one. First option.
Okay. Yes, this is the. I have. Next question is the. Oh, this is the same question. We'll discuss later. Okay. So, what was the answer given? It is given 2 and 3 only. Fluorescence quenching is observed in the room. This is the answer is given. Let's clarify because they are giving another answer here.
Yes, ma'am.
So this also we have done today. The match the following part. No need to do it again. The last question is carbon nanotubes. This one. So the assertion question is carbon nanotubes are highly soluble in water. Reason functionalization of carbon nanotubes has no impact on its water solubility. So I think carbon nanotubes are highly soluble in water. This is a wrong statement. And the functionalization of carbon nanotubes has no impact on its water solubility. It has impact. So both A and B are false. It should be. Yes, both A and R should be false. This is the last option. Okay. So this is not. This is now for today. So you just share. You can also share your mail ID. I think. Pratham.
Yes, ma'am. I'm
Yes.
just sending you the mail. I'm
Okay.
almost done. Just a second.
Okay. So we'll connect on the mail and clarify. So
I sent you the mail, ma'am. Please check
okay.
once, ma'am.
Just it hasn't come yet. Maybe it will come. This D dot studio that is fine. We can correct. Can you send again because it is yes, come. It has come. Yes. Okay. Okay.
Yes,
I
ma'am.
will see and will clarify on that. Okay. So it's
Okay,
for
ma'am. Thank
today.
you.
Yes.
Thank you,
Thank
ma'am.
you. Welcome. So I hope
Thank
yes,
you so
yes.
much,
I
ma'am, for
hope
your
you
time.
will be understanding something from this lecture, and it will be helpful for your doing the assignment. And if you have any doubt, you can contact. Also, you can see the video later also, because I will be uploading it in the YouTube, and I'll be sharing the slides with you in the Google Drive, so you can check from there also. So thank you for the class. Thank you for the listening to me. Till next Saturday. Goodbye.
Thank you.